Afternoon, everybody, and thank you to the organizers for having me here today. I think this is 25 plus years that I've been involved with this organization, nearly the beginning, so I'm getting younger. I don't know how that works, but I appreciate being here, and this is always one of my favorite medical conferences of the year because the ability to interact with my colleagues in science and in medicine, with patients and with families is incredibly unique, and thank you for all of the questions that you've asked so far. My goal today is actually a lot easier than all of the preceding speakers because I'm going to just tell a story about development so you can understand what it takes to actually get one of these trials done and ultimately, hopefully, get a new treatment for you. And some of what I'll be talking about, you might have already heard from the previous speakers, including Dr. Marmorelis, who gave a great overview of many of the trials that we had done when I was at the University of Pennsylvania. I'm just telling you a little bit about a story about how we develop trials and then a little bit about how to interpret the studies. When you're reading and hearing about them, it's important to be able to discern, especially if you pick up a medical journal and trying to read that medical journal and trying to figure out what it's trying to tell you. There's a lot of nuance there that's really important, some of which you've heard already today. So these are my disclosures, nothing directly related to this specific talk. But just an idea, when you're thinking about clinical trials and enrolling in clinical trials and thinking about drug development, you're hearing about this new thing that's in a mouse and you can't wait to have it that you can use for yourself or for your loved one. Remember that the timeline from first concept for development of a new drug or a new agent, a new therapy, to actually getting to the patient for routine clinical use is incredibly long and complicated. And it's longer if you're initiating this process at a university, at a medical school, than it is in industry, in a pharmaceutical or biotech company, but it's still, in either way, it's a long time. And the process, we hope, is going to get shorter over time, but it is long. And so we ask your patience with us as we go through this process. And just as you're thinking about the different types of studies, it's important to understand the nuances and the size of the study and the timeline in which these take. So we hear about phase 1, phase 2, phase 3. Some of you may hear about phase 4 clinical trials even. And remember that a phase 1 trial is a small study, generally in mesothelioma, probably closer to 20 patients or maybe even fewer. Takes several months to a year just to enroll all the patients in that study, maybe sometimes a little bit longer. And the primary purpose is safety. Is this new drug that we're testing, is it safe? Phase 2, maybe more. In mesothelioma, probably 50 to 100 patients, maybe. It may take a couple years to get that phase 2 study done. And we're looking at some safety, but mostly you're looking, does this work? Is this effective? And then phase 3 in mesothelioma, probably closer to a couple hundred patients, not 1,000 to 5,000. Those are more for hypertension studies or diabetes studies, but they take several years to do a phase 3 trial. And this is where you understand whether something works definitively. And this is where things can be really disappointing. You may see a really promising phase 2 study being published in your local newspaper, and that doesn't necessarily mean that as it's translated to a phase 3 study that efficacy is going to be shown. So you'll see many things that work in the phase 2 setting, but when they expand it in phase 3, they're unfortunately not very effective. So the problem, why are we all here today? We're here because we haven't solved the problem of really effective, safe therapies for mesothelioma. We are far better than when I first came to this meeting 25 years ago, but we have a long way to go. So what do we need to do? We need to do research and development. This starts in the laboratory, and that laboratory could be at a medical school, it could be at a university, it could be at a biotech firm. But it requires thinking about, what is the nature of this cancer? What does it make up? What are the genetic abnormalities? What are the predispositions? And how can we attack it, right? So you've got scientists and doctors thinking about these problems all the time, and then thinking about, well, what are the ways to do it? So you heard from Dr. Marmorelis that years ago, again, 25 plus years ago, when I was training at the University of Pennsylvania, it was the early days of being able to manipulate the genes of cells. And so we were really thinking about, could we manipulate the genes of cancer cells to make them die, and specifically mesothelioma at that time. And so just note the date of this paper that we published, 2001. So this is 23 years ago. We published our first paper on a mouse model showing that we could give a gene to a mouse that had mesothelioma, and we could cure that mouse of mesothelioma. That red line at the top are mice, that's survival, 100% survival of mice that receive this modified cold virus expressing this gene called interferon. And so in this model, again, curing a mouse is very different than curing a human, but you got to start with helping mice first to see if they can live. And so this was 23 years ago, just to keep that in mind in terms of, and that's when you published it. The experiments were done in the couple years before that. So then if you have something that's promising in a mouse, then you have to go to the FDA. And you have to say, we would like to take this novel compound. And back then, we were talking about genetically modified viruses. So this wasn't a simple, oh, we have a small molecule or a protein. We were talking about delivering viruses to human beings, which were genetically modified. So there was a long conversation with the FDA. And you have to put in what's called an investigational new drug application, or an IND, which by itself, the one we put together was about this big, multiple hundred pages in size, a year's worth of work just to submit the application to do the first in-human clinical trial. This was done by my primary mentor, Dr. Stephen L. Belda, and then we were able to get approval to go ahead with the first phase 1 study with this. And again, remember what the phase 1 study is. You'll see it in bold. What are the safety issues with the new drug? What is the optimal dose? And is there enough of a signal to proceed with additional studies? And understand that we're hoping to see some glimmer of hope in a small number of patients, 15, 20 patients, enough that it will justify going forward. So this is what a phase 1 study is. A phase 1 study does not definitively tell us whether something works or doesn't work. It just tells us whether it's safe, whether we have the right dose, and do we have enough information to go forward. And so then we developed a series of early phase clinical trials that we published 2007, 2010, 2011, because we were tweaking the virus itself and the doses and the time interval of delivering. And each trial took a year or two, and then we had to analyze the results and then put it together and then publish it. And again, all we're showing at this point is, what is the correct dose? What is the sequence? And is it safe? And do we have enough of a signal to go forward? Because if you don't have a signal, it can be perfectly safe, but if we don't see any benefit to patients, we're not going to go forward to the phase 2 study. So we saw this. So this was a PET CT scan of a patient who had been through surgery with mesothelioma, had recurred in the right side of the chest. You may see circles on the left panels. All those areas, circles surrounding white dots, are areas of recurrence after a pleurectomy, decortication, chemotherapy. Immunotherapy with checkpoint inhibitors didn't exist at the time when we were doing this. And that in this patient, a single dose of the virus injected into the chest cavity with no other treatment within 6 months almost led to a complete response. So again, this was not every patient, but this was one patient. And in this one patient, it was enough that it said, okay, we can go to the FDA and get justification for a phase 2 clinical trial. Again, this is 10 years after that first study in a mouse. So then we went to a phase 2 study. This is a moderately sized study that preliminary defines the effectiveness. Again, we're hoping that if we do more patients and we study this better, that we can assess how effective this is and hopefully have even more of a signal so we can go to a phase 3 clinical trial. Now, there are some phase 2 trials which are definitive enough and large enough that you might be able to skip the phase 3, go directly to the FDA, and be able to get approval for that drug that you're studying. One of those, for example, is tumor treating fields. I saw someone who was walking around with their tumor treating fields here before. That actually, a large phase 2 study was sufficient to go to the FDA. Actually, it was a single arm phase 2 study. They were able to get that approved. So there are some situations where you can skip the phase 3 portion. But this was a very complicated phase 2 study that we designed and published 2016, 15 years after the initial mouse study was published. And this is where we combined the delivery of the virus into the chest cavity, 2 doses, with an anti-inflammatory drug, celecoxib or Celebrex, with chemotherapy, either for patients who never received chemotherapy before or patients who had failed frontline chemotherapy and then were getting second-line chemotherapy. And what we hoped would be that we would see benefits in both. First line and second line, there would be addition. Notice that although we have two different groups, combinations in first line and second line, that we don't have this is not a randomized study. So there was no one who just got chemotherapy in this trial. So this is a stratified single-arm trial with no randomization. So this is a classic phase 2, although we did a little bit of a safety, so it's a phase 1-2 because we looked a little bit of safety of the combination first. And what we saw, this is the waterfall plot you saw from Dr. Marmorelis earlier. We showed that we had a number of patients that had responses, and this is the survival curve on the right-hand side, the Kaplan-Meier survival curve. And again, this was about double the historical survival for chemotherapy alone for patients in the second line. So it's not where we want to be, but at least compared to historical controls, showed us that we think we were improving survival. Now, the downside was we didn't show benefits in patients who had never received chemotherapy. So getting the first chemotherapy, that combination, for whatever reason, didn't work. So that was counterintuitive to us. We couldn't figure that out. We're still studying why that is. But we decided that it worked for patients who had been through chemotherapy, had progressed, give it in combination, and then we went back to the FDA at that point. Again, another year and a half of discussions with the FDA to design a phase 3 randomized controlled trial. And again, the goal of this was to determine definitively whether this was an efficacious treatment for patients with mesothelioma. So we designed this trial. This is the INFINITE study that you heard about this from Dr. Marmorelis. This was a global clinical trial which randomized patients who had been through frontline chemotherapy to getting virus plus chemotherapy plus the anti-inflammatory drug versus chemotherapy and the anti-inflammatory drug. So this was a randomized controlled trial. It was called open label because the people who got the virus injections, you knew you got a virus injection because you had a procedure to have it put in. So it wasn't blinded. People were aware of what they received. And this was, again, a global study with multiple centers across the world, including 11 centers in Russia, which is important as we think about this. Now, the timing of this study, and timing is everything in life, is that the first patient enrolled in this study, which I'd been spending most of my career developing, was August 2019. Little do we know that 6-7 months later, a global pandemic occurred. So when you heard that the study was closed, it wasn't closed because of lack of efficacy. The study was closed because we couldn't get people into clinical trials because many clinical trials in cancer and other diseases were slow to get people in because no one wanted to leave their house in the middle of one of the worst global pandemics in the history of humankind. So it closed in the spring of 2021 after randomizing 53 patients in the study. Again, this was the first ever randomized controlled trial of a gene therapy in patients with mesothelioma, but yet we are going forward in analyzing the data from those patients. And I'm hopeful that now that the pandemic hopefully is over and no further pandemics are coming, that we can consider revisiting this because perhaps combinations with immunotherapy may be even better. So in the next few months, we'll be analyzing the data from these randomized patients. I'll be publishing the data and presenting it hopefully next year at this conference. I can show you what the data shows. And hopefully, if there's a signal, we can go forward and hopefully work to getting to this to patients. I'm going to skip through the next slides just because of time, but just to say that once you go through that process, once you go through that phase 3 trial, then you have to put through a new drug application called an NDA. So you have to take all of the data that you accumulated from your phase 1, phase 2, phase 3 trials, and your preclinical studies in mice, and put that all together and submit that to the FDA. And you hope that the FDA will approve it. And just because you've shown efficacy in your phase 3 trial doesn't mean that the FDA automatically is going to approve this for use in patients. There's no guarantee of that. So just think about that process. We started these experiments 25 years ago. We're now analyzing the data from the phase 3 trial, which was curtailed by an unanticipated global pandemic. And we're hoping we can get to a new drug application that we can get this to you to use in your patients now. So I really thank you for your time and your patience with us in this clinical trial development and understanding that we really want to get these therapies to you as much as we possibly can. Thank you so much.
Thank you, Dr. Sterman. I see one question for Dr. Sterman, and I know he's headed out to the airport shortly, so we'll take a couple of questions if we can.
Thank you very much, Doctor. Is that on? Can you hear me?
I can.
Okay. You referred to a number of things here, including the pandemic and so forth, and the length of time, generally what it takes to get a new drug into the general population for treatment. I guess my question is this. It seems to me that when that pandemic occurred, there was a fairly terrific acceleration of drug testing and experimentation to get those drugs that would take care of the COVID virus onto the marketplace. So my thought is, however uninformed I am, is that there are two major components to the development of a new drug and finally getting into the population. There's the scientific time and activity that has to go through it, and then there's this bureaucratic time with the FDA and all the things you have to go through with that. Do you see from the COVID process any opportunities to streamline this current process that you've described for any new drug?
I think that's a great point. So you're referring to Operation Warp Speed, which helped really saved millions of lives worldwide. We heard about the mRNA vaccines. I was head of pulmonary and critical care at a hospital in New York City where we were absolutely inundated. It was apocalyptic in New York City at that time. And without those vaccines, I can't even imagine that our hospitals and our health systems would have survived. The miracle is that the pandemic hit in February, March of 2020, and we had vaccines rolling out by December of that year. Absolutely remarkable. But this was a concerted worldwide effort. Everyone focused on one biomedical intervention, right? So that's, I would love for everything to be able to be rolled out in 10 months. We can get this to you. But this was an absolute global imperative with multiple billions of dollars poured in to this one initiative that the world was focused on. But are there lessons that we can learn? Yes. First of all, take for a grain of salt the timeline that I gave you. The timeline is for what's called investigator-initiated clinical trials. I was a junior faculty member. This was a concept I developed with my mentor. We're trying to get grants approved through the National Cancer Institute. Very different timeline than in industry, than in biotech, than in large pharma, where they've got hundreds and hundreds of people working on a problem, not just me and a postdoc in a laboratory sitting in Philadelphia somewhere. So again, industry has a more accelerated timeline than I do. But I think that your point is well taken, which is if there's enough initiative, this is where advocacy can play a major role. You can go to FDA and say, we want acceleration of development. The classic example of that is HIV drugs, right? In the 1980s, the community came to the FDA and said, people are dying. We need to fix this problem faster than you're acting. And Dr. Fauci, to his credit, agreed with them and helped accelerate drug development. So this is where this foundation can play a major role in accelerating this process. Thank you very much.
Thank you, Dr. Sterman.